Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Luqman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the module Lipids, Inborn Errors of Metabolism from the paper Lipid Metabolism. The objective of the present module is to understand the lysosomal storage disorder with respect to inborn errors of metabolism. What are the plausible causes of lipidoses and how many types of lipidoses occurs? First, let us discuss the lysosomal storage diseases with respect to inborn errors of metabolism. The lysosomes, an organ which was discovered by de Duve in 1955 and is the organelle credited for recycling and breakdown of cellular components. The physiological turnover is mainly caused by the catabolic enzyme working at low pH within the lysosomes. Hindrances in the function ultimately leads to an accrual and storage of components which leads to the various clinical abnormalities and death. The lysosomal storage diseases in general is the collection of hereditary metabolic disorders caused by the flaw in the protein responsible for the proper functioning of the lysosomes. Most of the lysosomal storage diseases are fatal. The major abnormality includes ocular pathology, organomegaly, connective tissue pathologies, and mental retardation. The neurological problems of the lipidoses mainly are brain degeneration, ataxia, pain in the arms and the legs, seizures, eye paralysis, learning troubles, swallowing in the food, difficulties, spasticity, and failure of the muscle tone. There is a slurred speech and hypersensitivity to touch and clouding of the cornea. It is now accepted that these disorders are not simply an outcome of storage, but an end result of perturbation of the multifaceted cell signaling pathways, which leads to the specific structural and biochemical changes which can provide the hint for the therapy, but still there exist challenges for treating the effects on the central nervous system. The main types of enzymes includes the enzyme responsible for catabolism like hydrolase, post-translational lysosomal enzyme modification defects, metabolite proteins important for appropriate intracellular trafficking, mainly in the trans-Golgi network, which involves the lysosome and the activator proteins. The enzymes specifically responsible with respect to lipid metabolism are discussed in this module. They are caused by defects in proteins that are critical to the functions of the lysosomes. The concept map of a cell. This picture represents a cell with different organelles, and especially because we are focusing on the lysosomes, we can see here the primary lysosomes, the nucleus, nucleolus, Golgi apparatus, the rough endoplasmic reticulum, smooth endoplasmic reticulum, and we can see here the precursor enzymes of the receptor separated and enzyme delivered to lysosome for final activation, and then receptors return to Golgi. The inactive enzyme acquire address for lysosomes and bind to the receptors. These vesicles containing enzymes bud off from the endoplasmic reticulum and travel to the Golgi apparatus. You can see here the ribosomes on the rough endoplasmic reticulum synthesis. These synthesize the inactive precursor enzymes, which further brought to the lumen of the endoplasmic smooth surface receptor, and then vesicles containing enzymes bud off from the endoplasmic reticulum travel to the Golgi apparatus, and from these Golgi apparatus, the inactive enzyme acquire address for lysosome and bind to the receptors. Then these receptors separated, an enzyme delivered to the lysosome for final activation, and receptor get back to the Golgi apparatus. Lipidoses. Lipidoses, or the lipid storage disease, are a collection of hereditary metabolic inborn errors in which the detrimental quantity of lipids, or the fats, mount up in various of the body's cells and tissues. The lipidoses are inborn metabolic errors leading to the accumulation of various lipids in the nervous system and in the reticuloendothelial system. These metabolic disorders can occur because of mainly two reasons. First is that enough enzyme production or improper functioning of the enzymes, and therefore the unnecessary storage of fats can cause everlasting cellular and tissue harm, chiefly in the peripheral nervous system, brain, liver, bone marrow, and spleen. Lipids are generally distinct as any naturally occurring fat soluble, that is lipophilic, such as oil, fat, steroids, Paxes and others. And lipids are the vital parts of the membrane found within and between each cell and in the myelin sheath that coats and guards the nerves. Lysosomes are the main organelle which metabolize and converts lipids and proteins into smaller parts for energy production. Lipid storage diseases are related to diverse disorders which are hereditary, mostly occurs due to deficiency of lysosomal enzymes. The major types of lipidoses are the lysosomal storage diseases. This is a kind of lipid storage disorder, and it can be seen in a triangular form. Mainly, at one end, it is sphingolipidosis. The other end, the neuronal ceroid lipofuscinoses, and finally, acid lipase disease. The lipidosis or the lipid disorder can be well understood under these three disorders. The inheritance pattern of lipidosis. The lipid storage diseases are hereditary, in which defective gene regulating a specific protein carried by one or both parents, and these are inherited in two ways. The autosomal recessive inheritance that happens when both parents transmit and convey a copy of the defective gene, but neither parent is influenced by the disorder. Only 25% of the children can be affected by an autosomal recessive inheritance. In this picture, you can see that father is the carrier and mother is the carrier, which is an autosomal recessive. And you can see that only one is affected out of the 4 offspring. 2 were unaffected carrier and 1 is unaffected non-carrier. The second trait, that is X-linked or the sex-linked recessive inheritance, happens on the X chromosomes. The mother carries the targeted gene that decides the child's gender and passes it to her son with 50% possibility in the carriers of the disorder inheritance. Types of lipidosis. The lipidosis can be of many types, and the first and the foremost is the sphingolipidosis, and these are the sphingolipid storage diseases. Essentially an autosomal recessive disease, except the Fabry disease, which is X-linked. Broadly, sphingospholipids and glycosphingolipids are the main groups of the sphingolipidosis. Ceramides are the precursors of both the phosphorylated and glycosylated sphingolipids. Under these sphingolipidosis, sphingospholipidosis occurs. And the most important sphingophospholipid in human is sphingomyelin. This sphingomyelin is an important constituent of the nerve fibers, myelin of the nerve fibers, and have a structure in which an alcohol group at carbon 1 of sphingosine is esterified to phosphorylcholine. The ceramide is the immediate precursor of the sphingomyelin, and it is the major constituent of skin and regulates the skin's water permeability. Sphingomyelin is degraded by sphingomyelinase, which is a lysosomal enzyme that removes phosphorylcholine and leaving behind ceramide. The ceramidase cleaves ceramide into sphingosine and free fatty acid. Ceramide and sphingosine regulates many signal transduction pathways, mostly PKC dependent pathways, and also promotes apoptosis. The Niemann-Pick disease is actually a group of autosomal recessive disorders caused by its inability to degrade sphingomyelin by the deficient sphingomyelinase enzyme. There are two forms, the type A and type B, or it may happen due to the lack of NPC1 or NPC2 proteins, which also occurs in two forms, type C and type D. The sphingomyelinase is a type of phospholipase C, and the liver and spleen are the primary sites of deposition of lipid, while the deposition of sphingomyelin in the central nervous system leads to progressive neurodegeneration. In this, there is one disorder which is called as glycosphingolipidosis, where the transformed cell with dysregulated cell growth and division leads to a dramatic change in the glycosphingolipid composition of the plasma membrane. They are antigenic, that is, the blood group antigens or the embryonic antigens specific for the particular stages of fetal development and tumor antigens. Glycosphingolipids consist of neutral and acidic types. Further, within acidic glycosphingolipids, there are two types, gangliosides and sulfatides. The main features of sphingolipidosis diseases are as follows. The GM1 Gangliosidosis. In this, there is an accumulation of gangliosides, GM1 and keratin sulfate, as a result of which there is a neurologic deterioration, there is hepatosplenomegaly, there is a deformities in skeletal muscles, and cherry red macula. In Tay-Sachs disease, there is accumulation of ganglioside GM2, which causes rapid, progressive and fatal neurodegeneration. Also blindness, cherry red macula, muscular weakness and seizures occur. Gawker's disease. Gawker's disease is characterized by the accumulation of glucocerebrocytes. It is the most common form of lysosomal storage disease, which is characterized by the hepatosplenomegaly, osteoporosis of long bones, and involvement of the central nervous system in rare and infantile and juvenile forms. Sandoff disease. Sandoff disease is characterized by the accumulation of GM2 and globosides. This disease gives the symptoms similar to that of Tay-Sachs disease, but in addition with the neurological symptoms, it gives visceral involvement as well. Fabry disease is another disease which is characterized by accumulation of globosides only, and as a result of the accumulation of globosides, there is a reddish purple skin rashes occurs. Severe symptoms include kidney and heart failure and burning pain in the lower extremities. Metachromatic leukodystrophy. It is the disease characterized by the accumulation of the sulfatides, which shows the symptoms of cognitive deterioration, demyelination, progressive paralysis, and dementia in infantile form. Nerves stain yellowish-brown with crescent violet, metachromasia, and multiple sulfatase deficiency due to defect in post-translational modification of several sulfatases. The Niemann-Pick disease is characterized by the accumulation of sphingomyelin, as a result of which there is a hepatosplenomegaly, there is a neurodegenerative course of type A, and cherry red macular appearance occurs. In globoid cell leukodystrophy, which is also called as Krabbe's disease, the accumulation of galactocerebrosides occurs, which causes the motor deterioration as well as mental deterioration. The severity of the disease causes blindness and deafness, and there is a near total loss of myelin. Also, globoid bodies in white matter of brain has been lost. In case of Farber's disease, there is an accumulation of ceramide, which causes painful and progressive joint deformity, and subcutaneous nodules of lipid Leydig cells. There is a hoarse cry, and tissue shows granulomas. The acid lipase diseases or disorders. The flaw in the acid lipase enzyme, which is required to degrade mainly fatty acids, oils, and cholesterol, resulting in the accumulation of these moieties. Two rare inborn lipidoses are because of lysosomal acid lipase deficiency. One is cholesterol ester storage disease, CESD, and another is Wolman's disease. Wolman's disease is an autosomal recessive disorder, lipid storage disease, in which cholesterol esters and triglycerides are accumulated. The child appears normal at birth, but quickly develops progressive mental deterioration. The hardening of adrenal glands because of the calcium deposits takes place, and enlarged liver and spleen with gastrointestinal problems like anemia, jaundice, and vomiting occurs. There is a low muscle tone in Wolman disease. In the cholesterol ester storage disease, which is an exceedingly uncommon disorder, where cells of blood and lymph accumulate cholesterol esters and triglycerides. Diagnosis is not an easy process in this case, as the onset fluctuates and sometimes the diagnosis occurs at a later stage during adulthood. The symptoms include the enlarged liver, and problems associated with it like cirrhosis, and thus before adulthood chronic liver failure most probably occur. Jaundice as well as calcium deposits occurs in the adrenal glands. The third form of the lipid storage disorder is neuronal ceroid lipofuscinosis, which is also called as NCL. The neuronal ceroid lipofuscinosis results from the unnecessary accretion of the lipofuscin, which is a lipopigment. And these lipofuscin are made up of colored proteins, actually greenish yellow color when viewed under an ultraviolet light microscope. And lipids in the body tissues, mostly in the neuronal cells, while it is also present in the myocardium, spleen, liver, and kidneys. The main types described in the following figure. The types of neuronal ceroid lipofuscinosis include the infantile neuronal ceroid lipofuscinosis, late infantile neuronal ceroid lipofuscinosis, juvenile neuronal ceroid lipofuscinosis, and adult neuronal ceroid lipofuscinosis. These four types are also called as Santivori-Haltia disease, Jansky-Velikovsky disease, Batten disease, and Kuff's disease respectively. The first, the infantile NCL, begins about 6 months and 2 years of age and progresses rapidly. In affected children, they fail to thrive and abnormally small heads, which is called as microcephaly. Whereas in late infantile NCL, which begins between the 2 and the 4 years, the typical early signs are loss of muscle coordination, which is called as ataxia, and seizures, along with progressive mental deterioration. The affected children may show mild severe delays in speech development well before other symptoms appear. This form progresses rapidly and ends in deaths between age 8 and 12. The third form, which is juvenile NCL, begins between the age of 5 to 8 years. The typical early signs are progressive vision loss, seizures, ataxia, or clumsiness. This form progresses less rapidly and ends in death in the late teens or early 20s, although some have been known to live into their 30s. Generally, begins before the age of 40. It causes milder symptoms than progress slowly and does not cause blindness. Although age of death is variable among affected individuals, and this form does shorten life expectancy. The diagnosis of lipid storage diseases. The diagnosis of lipid storage disease is possible through various modes. The first mode is the clinical examination of the urine, the analysis of the urine, and discovering the existence of the stored material. Second mode is the genetic testing and the molecular enzymatic analysis of the cell, of the affected tissues, or blood, or any other body fluids. The next mode may be genotyping, in which some specific test can be done to verify the person carrying the defective gene that can be passed on to her or his children. Biopsy for the lipid storage disorders involves the study of cells and tissues like liver by removing a small amount of tissue done through surgery or by needle biopsy. The genetic testing mostly prenatal testing, is done during early pregnancy through chorionic villus sampling, where little placenta tissue sample is removed and tested. This can help in detecting lipid storage diseases of individuals who have a family history. The prognosis and the treatment of the lipidosis. The prognosis for a lipidosis is determined by the kind of illness, by the severity of the symptoms, and also the age of the commencement of the disorder. Children with Niemann-Pick disorders, they mostly die at an early age with problems like progressive neurological loss or infection, while children with Gawker disease or disorders may live healthy up to adulthood. Most children having Farber's disease die with lung problems before the age of 2, as well as children affected with Fabry disease often die prematurely of complications from renal failure, heart disease, or stroke. The Tay-Sachs disorders may ultimately want a feeding tube, and thus they often die by age 4 from chronic infection. At present, there is no precise treatment available for most of the lipidosis or the lipid storage diseases and disorders. Like that of Niemann-Pick disease, even though Fabry's and Gawker disorders have extremely successful enzyme replacement therapies to relieve the pain in Farber's disorders, doctors often advise corticosteroids, while in Tay-Sachs disease, to control seizures, anticonvulsant medications are often prescribed. So students, in this module, we have discussed about the lipid storage diseases, the types of lipid storage diseases, the disorder of the lipidosis and its various form, the diagnosis, the prognosis, as well as the treatment of the lipid storage diseases, lipidoses, and their related disorders. Thank you.